So in the previous video, we were talking about the AC and DS elements in maze, and the video stopped in out of nowhere. So we have to continue with this topic, and we were talking about the AC and DS elements. So let us try to first learn the basic concepts of the same, that what is the meaning of AC and DS elements, and then we will move on further. Okay, so basically AC and DS are the elements which are the parts of the transposons, and these transposons are located inside the genome of the maze, specifically the kernel of the maze. Now AC and DS elements full form is AC stands for activator transposon and the DS stands for the dissociation transposon. So one of them is activator transposon and the another of them being the dissociation transposon. Now the two of the transposons are responsible for the pigmentation in the kernel of the maze. So now we have to talk about that how do they cause the pigmentation in the kernel of the maze. Before that, you should also know that there was a scientist whose name was Barbara McClintock, and she was the one who discovered these transposable elements in the maze. And it was a landmark discovery, and it has clearly proved that how the pigmentation can vary in the kernels of the maze. Just have a look on the particular pigmentation, how this pigmentation is occurring in the kernels of the maze. You can see that whole of the maze kernel is otherwise yellow, but there are the spots of the pigments present on it. Now these spots of the color are referred to as sectors, or they are also called as streaks. So they are called as sectors or streaks which are present on the maze kernel. Now these streaks, sometimes they appear, sometimes these disappear. It is all because of these transpositioning of the elements AC and DS. So now what happens? Let us have a look on these particular elements, that how do these elements look like. So here is your AC element and here is your DS element. The AC element is a full-fledged transposon which consists of this pink colored FDR region, then ITR regions, these green colored, and here is the gene which is coding for the transposase enzyme. The DS element is the modified form of AC element. It consists of DS9, DS2D1, DS2D2 and DS6. All of these particular genes or all of these particular elements, they have one of the lacking thing, or they have deletions in their transposase gene. It means that the DS elements cannot produce their own transposase enzyme. Now mainly for the pigmentation in the kernel of maize, the DS elements are responsible. But these DS elements first have to move from their original place and have to be inserted into that particular gene which causes the pigmentation. So that pigmentation gene in the maize is C gene. Now this C gene can have two alleles, capital C and small c. This capital C is the dominant allele and it will cause the pigmentation. Now in the case when the kernel has capital C genotype, the coloration will be definitely produced, or the streaks will definitely appear. If there is a small c, that time it is the yellow kernel or it is the colorless kernel. And when it is capital C small c, that time the interference of AC and DS elements will take place. Now what happens is this C gene, along with this only, in the continuation, in the same genome there is AC element also, and then there is DS element also. The AC element, when coding for transposase enzyme, this transposase enzyme works on the DS element and will cut it or cleave it from the FDR region or the ITR regions. This DS element will then move, and then afterwards will have here on the C gene also the transposase enzyme will work, and the C gene will be cut out in from between. The DS element will be inserted over there. Now as soon as the DS element is inserted on the C gene, it becomes CTC. The T stands for transposon. which has come here. Now once this T is inserted into this capital C gene, the coloration stops and the kernel becomes yellow in color. Later on, because these transposons are mobile elements, the excision of the particular transposon will happen. The excision of DS will take place later on, and as soon as it is removed, it will again become the normal capital C small c genotype, and that time the pigmentation will reappear. Now because of this continuous jumping in and jumping out of the transposon from this C gene, the pigmentation will appear, then will not appear, will appear, will not appear. In that condition, we see that on the kernel of the maize, there are these streaks or the sectors of purple, then yellow, then again pigment, then again yellow. So it's like uh, pigmented, not pigmented, pigmented, not pigmented. It is all because of this transpositioning nature of the DS element. So this is how the pigmentation takes place in the maize. Here the diagram has also been clearly explained. So suppose if this is your normal kind of phenotype wherein you have small c and small c. In this condition, no matter what, always the yellow kernel will be produced because here you have small c, small c. Now in the case if you have capital C here, in this capital C case, the purple kernel will be produced. But this purple kernel, this purple pigmentation will not last long because the DS element will then be inserted into the C gene. As soon as this DS element inserts itself into the C gene, it becomes CT. When it becomes CT, the kernel becomes yellow. So this coming in and coming out of this DS element is causing the variegated kernel. Variegated kernel means the alternating or altering sectors on the kernel. Altering sectors of what? Altering sectors of pigments on the kernel. So the variegated corn kernels result from the excision of DS elements from genes controlling the pigment production during the development. So it all happens during the development only, that the DS element is inserted and later it is also removed. It is all happening during the developmental phase. Also remember, if the excision of this DS element occurs early in the development, means it is exiting early from that particular C gene, then the sectors will be broad, the pigment sectors will be broad and large. But if the excision is late in the development, only the few cells will have this functional capital C allele and the pigmentation sector will be small. So this is how the ACDS elements work in the maize. Now we have to talk about the transposable elements in Drosophila. So another important topic for today will be the transposable elements which are present in Drosophila. Now Drosophila consists of important transposable elements and they are commonly called as P elements. Although there are other elements also which we will talk about later, or you can refer here from this table. There is one copia element also in Drosophila, but we have to discuss this P element in Drosophila. Now the difference between the copia transposon and P transposon is that copia transposon is a form of retrotransposon, while this P element, it is here not the retrotransposon, it is just the class 2. Class 2 represents the normal transposons or the DNA transposons. So here we have to discuss about the P elements in Drosophila. So these P elements are present in the genome of Drosophila and they move from one place to another through the copy paste mechanisms. So let us come here. Okay, this is very important and you have to listen to it very carefully. So the P elements are approximately 2900 base pairs long. They move by copy paste mechanism, that is by the DNA. Sorry, not copy paste, by cut and paste. Means they will cut from this original location and then they will move into the target DNA and they will be pasted there. They do have the inverted terminal repeats, that is ITRs, and they also generate the short flanking direct repeats, which is universal feature of every transposable element. Now this mature P element or this mature transpose TE element, what will it do? This transpositioning element will generate or will encode for transposase gene, and this transposase gene will result into the mobility of the transpositioning element or the mobility of TE. This same P gene also codes for one more enzyme, that is the repressor enzyme or the repressor protein of this transposon. Now this repressor protein, as soon as it is coded by this P gene, it moves into the cytoplasm and it remains there. This repressor protein is helpful to stabilize this transposon and to increase the stability so that the mobility can overall be decreased and the chances of mutation can be decreased. So what I have told up till now is that this P element in Drosophila is actually a P gene which codes for two further subgenes, one of them being the transposase gene and one of them being the repressor gene. Now transposase gene in the P element will code for the transposase enzyme, which will help in the mobility of the transpositioning element, while the repressor gene in the P element will code for the repressor protein, and this repressor protein will move out into the cytoplasm and later it will help in achieving the stability to the transpositioning elements by decreasing its mobility. Now listen very carefully. Those individuals of Drosophila which receive both of them, or which receive in short the repressor protein, we are concerned about repressor protein. Why? Because if repressor protein is present, then the stability will be there and the chances of mutations will be less. But if the repressor protein is not present, the stability will be not there, and in that condition the chances of mutations, chromosomal deletions, additions, alterations would be very high. So what we are concerned here is about the repressor protein. If the repressor protein is available, that time we say that the Drosophila individual is P positive. And if the repressor protein is absent, that time we say that the Drosophila is P negative. So in this condition what happens? The P positive individual will consist of this repressor protein and the P negative individual will not have the repressor protein. Now let us see what is the significance of this. So moving on to the significance of this particular repressor protein, the important thing here is that those individuals which have P positive protein, or which have this which are P positive or which code the repressor protein, they are at advantage because their genome is comparatively stable. Now you think of a cross between P positive father and a P negative mother. Now because the repressor protein and its gene is present in cytoplasm, please remember that this repressor protein is always present in cytoplasm, so it is the condition of cytoplasmic inheritance. So through this cytoplasmic inheritance, the progenies will not have, or I can say that the progenies will be P negative. Why? Because the cytoplasmic inheritance always takes place from the mother. The mother's cytoplasma is passed into the progeny, and in that condition, if the mother is not having this gene coding for repressor protein, then the progenies will also not have the same repressor protein. So if the repressor protein is not present, the progenies will be very unstable and their genes will show very high chances of mutation, very high chances of instability, chromosomal aberrations, and even the sterility will be there. So this condition is referred to as hybrid dysgenesis, and it is very important to know in the peculiar case of Drosophila. Now let us move to the transposable elements in humans. Now transposable elements in the humans have now, most of them have now become inactivated and very less of them are actually active. And this is one of the reasons that why human genome is comparatively very much stable. But there is one interesting fact that almost 45% of the genes which have been derived in the or which are present in the human DNA have been somewhere directly or indirectly derived from these transposable elements. Although these transposable elements now have become inactive, but there was a certain time at which or during which these transposable elements were very active, and they had encoded for very new kind of genes and innovations, and which have resulted into the formation of these new genes and which are present in our DNA. So approximately 45% of the content of DNA is derived from these transposable elements. Moving on further, uh, if I talk about 5 to 6 million years ago, these transposable elements have played a very important role in humans. Actually, humans, monkeys, chimpanzees, apes, all of them are primates. And in these primates, approximately 5 to 6 million years ago, there had been 11 to 12,000 transpositioning events that took place inside the genome of these primates approximately 5 to 6 million years ago. Because of that, the evolution took place and the genome had diversified, and because of the genome diversification, some of these primates evolved into apes, some of these primates evolved into humans, some of them evolved into the monkeys. So this all diversification and evolution has been a resultant of the transposition events which had taken place approximately 5 to 6 million years ago. Now, as I was talking about the transposable elements in humans, one of the very common transposable element in human is ALU gene. This ALU is found frequently and abundantly in the human genome. Approximately 1 million related copies of ALU are present in one genome of one cell of human. Now the ALU sequences or the genetic sequence of ALU is very similar to 7S RNA molecule. This 7S RNA molecule is responsible for the transporting of the proteins through the endoplasmic reticulum. And the genetic sequence of this 7S RNA is very much similar to ALU sequences. So somewhere more or less we can derive that what ALU sequences might have played a role in. And that is up to you, and you should think about it. These ALU sequences are very short sequences, and because they are repeating again and again, so that's why we are saying that they are making so much amount of DNA, but it is all a repetitive form. These ALU sequences, when they insert itself into the DNA, they create short flanking direct repeats, and they have the characteristics that suggest that they have transposed through RNA intermediates. Means, in other words, we can say that these ALU elements somewhere in the history might have been retrotransposons. So these ALU belong to the class of repetitive sequences, and they are found in mammalian genomes. These are collectively called as SINE, which refer to as short interrupted nuclear elements, and they have comprised of approximately 11% of human genome, and they have been responsible for approximately 20 human genetic diseases. There are another class of such genetic sequences which are called as long interspersed nuclear elements, and they comprise of approximately 21% of human DNA, ALU being the part of SINE. Both SINE and LINE have a characteristic feature. When these are present in repeated form, suppose like this 5' to 3', their 5' end has been somewhat shortened, and this is the characteristic feature of both sine and line. So this is what about the repetitive sequences which are found in humans can be signs and lines. Now lastly we have to discuss about the significance of these transposable elements across all the organisms. One thing which we have very commonly understood is that these transposable elements are very important in innovation of the new genes. Since these transposable elements can move from one place to another and insert itself into a new gene, they can create a new separate gene. And this is what has led to the diversification into monkeys, apes, and humans. So, not only in this case, In the other cases, in other organisms also, this diversification and the innovation of new genes might have been created, all because of these transposable elements. Other than this, if we talk about, then these transposable elements even create mutations, duplications, deletions, all just to have the genome evolution. Also, one important feature is that under the stress conditions, these transposable elements become very active. And that time, due to certain mutations and chromosomal rearrangements, the new genes are made which encode for the proteins which will help the organism to survive during stress conditions. Now we have to talk about the characteristics of the two major classes of transposable genetic elements which will conclude this unit. One of them is class 1 transposons and one of them is class 2 transposons. Class 1 are the RNA mediated transposons, so they are called as retrotransposons. And class 2 transposons are the DNA mediated transposons. Class 1 transposons consist of terminal direct repeats, while the class 2 transposons consist of terminal inverted repeats. So ITRs, which we were discussing, is the characteristic feature of class 2 transposons. But in class 1 transposons we have terminal direct repeats. These terminal direct repeats in class 1 transposons are long, but the terminal inverted repeats in class 2 transposons are comparatively shorter. Then both of them obviously generate the short flanking direct repeats. The genes which are encoded by class 1 transposons include the reverse transcriptase. Why? Because their RNA has to again convert into DNA so that this can be inserted into the target gene. But the genes encoded by class 2 encode the transposase gene. This transposase gene is required to cleave the particular sequence of the target gene. Then the transpositioning in the class 1 transposons occur by RNA intermediate, but in the class 2 elements it occurs through DNA. The examples include the transposon yeast (TY) elements, then the copia elements in Drosophila and the ALU elements in humans. But the class 2 examples include IS1 elements which we discussed in E. coli, TN3 which we again discussed in E. coli, and ACDS which we again discussed in maize, and the P elements in Drosophila. So all of this have been covered, this has been covered, and this has been covered. Only the copia elements are left which you can study by yourself. So it is at your pace, you have to learn it by yourself. So this completes our transposons unit.